Hi, I'm Cole Carlson, and today I will be presenting a case of double trouble, two plastic-eating enzymes. The plastic that is being obliterated is called polyethylene terephthalate, or PET. It was invented by a pair named John Rex Winfield and James Tennant Dickinson in 1941. By the 1970s, it had become widely used, and today it is the most used plastic found in things from bottles, food containers, and fabrics. At the bottom here, you can see the basic esterification reaction that forms the PET polymer. We've all heard this story before. Plastic becomes popular with civilization. Civilization leaves plastic just laying about everywhere. Nature doesn't know what to do with it all, so it just accumulates. Now we have a plastic pollution problem, and all seemed lost. That was until 2016, when a team of scientists from the Kyoto Institute of Technology stumbled upon something interesting while looking through some trash from a bottle recycling plant in Sakai, Japan. They found a bacteria they named Ideonella sakaiensis after the town it was found in. I. sakaiensis was special because it had figured out how to eat PET. But how does it do this? It was proposed that the bacteria used a two-enzyme cocktail to break down PET. These two enzymes were called PETase and M-hetase, which, in concert, broke PET down into its component parts. On the left here is a schematic summary of the PET breakdown process. I'll go over what these acronyms are later. A couple years later, in 2018, a team was assembled to investigate these enzymes. The team was co-led by Dr. Greg Beckham of the U.S. Department of Energy's National Renewable Energy Lab, NREL, and Professor John McGeehan of the University of Portsmouth. They chose to first look at the enzyme responsible for the initial breakdown of PET, aptly named PETase, pictured here. The enzyme falls into the hydrolase family, and it's specifically a serine esterase. Structurally, what this means is that it has an alpha-beta fold that contains a catalytic triad at the active site on loop regions, and the nucleophile responsible for attacking PET in the triad is serine. We will be looking at the catalytic triad next. The catalytic triad is composed of serine, aspartic acid, and histidine found on loop regions at positions 160, 206, and 237, respectively. On the right you can notice the order and positioning of the three residues. Aspartic acid is in great position to hydrogen bond to histidine, which helps the histidine to depolarize the serine and prepare it for nucleophilic attack. Here is a bonus view of the active site. The team wanted to know more about this enzyme, so they mutated it and accidentally made it better. They made a double mutant by replacing the serine at position 238 with a phenylalanine, and the tryptophan at position 159 with histidine. The double mutant was found to be more effective than the wild type. The team concluded that this may be due to the PET substrate fitting better into the mutated enzyme, thus increasing binding affinity due to increases in surface contacts. It is also thought that it provides a better positioning for nucleophilic attack of the PET carbonyl. Unfortunately, the researchers did not publish a protein with these mutations that I could render in VMD, but pictured here it is the residues that would have been mutated. Pictured on the right is an example of a short PET chain, and depending on where the nucleophilic attack occurs, you can have three products form. You can have MHET, or mono 2-hydroxyethyl terephthalate, which is the major product. You can have TPA, or terephthalic acid, which is one of the initial components. It's only made in trace amounts, and it's usable to I-succiensis. Or you can have BHET, bis 2-hydroxyethyl TPA. Again, this one's only made in trace amounts, but it presents a bit of a problem because, as far as we can tell, there is no BHETase. As mentioned previously, MHET is the major product of the PETase reaction and one of the enzymes in the two-enzyme cocktail employed by I-succiensis is MHETase, pictured to the left. 2 years after accidentally improving PETase, the same team of scientists decided to look at MHETase. They found that MHETase was much bigger than PETase. Here's a size comparison. In addition to a hydrolase domain very similar to PETase, containing an alpha-beta fold with the same catalytic triad, MHETase also has a lid domain accompanied by a calcium ion. Here's a view of MHETase with the lid domain highlighted in pink and the hydrolase domain highlighted in light blue. Here's a view of the two domains separated from each other, with the hydrolase on the left and the lid domain on the right. The active site triad is just like that of PETase, composing of a serine, aspartic acid, and histidine at positions 225, 492, and 528, respectively. Again, on the right, we have a zoomed-in view of this catalytic triad, and you can see how the aspartic acid is very well positioned to interact with the histidine, which is also very well positioned to interact with that serine. These two enzymes are an example of convergent evolution. They don't really share a common ancestor that's very recent, but it's insane how similar their hydrolase domains are. The MHETase looks like it's just an extended alpha beta fold of the PETase, and the catalytic triad is even in the same kind of position. The lid is a bit mysterious because its role is not quite clear. It's stabilized by a calcium ion, shown in pink, and it is thought that the domain serves to stabilize the catalytic activity of MHETase. To test this, the team identified an analogous region in PET, shown on the right, and they swapped that loop region with the lid domain from MHETase to see what would happen. When they did this, neither enzyme wanted to work. It appears that MHETase really likes its hat, and PETase hates hats. In the graphic, I highlighted the lid domain on the left in yellow, and the loop region from PETase on the right in yellow as well. The green dots signify the spots that the researchers snipped the enzyme at and swapped the enzyme, or swapped the regions, and the orange blobs represent the catalytic triads. So now that I've gone through both enzymes, I just want to bring back that schematic from earlier again, and we can see that once PET has been broken down into B het, M het, and TPA, M hetase acts on the M het and breaks M het down further into more TPA and into ethylene glycol. And both ethylene glycol, e.g., and TPA can be used by isacaiensis. So after the team had characterized M hetase, they noted that the two enzymes work together in the wild. So they wondered if they made a chimeric version of the two enzymes, would it work better? And once again, this team improved the system's ability to break down PET. Good job, guys. To do this, they made a flexible glycine-serine linker and connected the C terminus of M hetase to the N terminus of PETase. Interestingly, it does not work if it's the C terminus of PETase connected to the N terminus of M hetase. Professor McGeehan is quoted as saying that the hybrid enzyme looks like two Pac-Men joined by a piece of string. Personally, though, it reminds me more of two chain chomps from Mario stuck together. Some future thoughts. Beyond the obvious uses for dealing with our plastic problem, these enzymes offer insights that could be used for synthetic enzyme design in the future. Like, we could use what we learned from how these enzymes break down PET to maybe engineer an enzyme that breaks down B het or other plastics. Maybe one day, as we develop new plastics, we also develop designer enzymes that are made to break down those plastics at the same time. Another thought would be like, what are the ecological impacts of using these enzymes to break down PET? Or what would the ecological impacts of using isacaiensis at landfill sites be? What would happen if these bacteria or these enzymes got out of a landfill environment and into like other areas? What would their impacts be on other like animals, or what kind of downstream effects should we be like looking out for if we were to employ this method to clean up plastic pollution? Could we engineer bacteria like Sacaiensis to break down microplastics in places like deep ocean floors or in our bodies? All in all, it's really amazing how PET has only been in mass use since the 1970s, and already these little guys have figured out how to eat it.